right, thank you very much, Manish, for agreeing to give a Build a Cell seminar, and you can get started.
Thank you, Kate. Thank you for the invitation. It's a pleasure to be here at this seminar series, which I, the entire group I try to follow a lot because you guys do a lot of exciting stuff building synthetic cell. And what I do is not quite in that direction yet, but we try to do small things that might contribute towards building cells. So with that, I'll start talking about the topic for the day, which is I'm going to discuss how to use linear DNA templates in exonuclease-deficient cellular extracts. My name is Manish Kushwaha. I'm a PI in the team BioRetrosynth. So in the French system, very often we have multi-PI teams. So the team BioRetrosynth has Jean-Luc Folland, who's the head of the team, and he's the main head PI. But then we have other projects that have other PIs, and I started at the Micalis Institute in 2017. The institute is located in the national lab called INRAE, which is essentially the French national lab for agriculture, environment research. And we're academically affiliated to the University of Paris-Saclay, which is in the south of Paris, as you will see. Yeah, okay. Right, I will tell you a little bit about my journey, as it were. So I grew up in a small town called, well, it's not such a small town, but in India, it's not the biggest town, let's say, right? So by standards of the world, it's quite a big city. I grew up in a town called Lucknow. Here I went to high school, up to my high school, and then from my university, I moved to New Delhi, where I did my undergrad and master's. And after that, for my PhD, I moved to Oxford, where I did my PhD, and I had a short one-year stint in the same lab where I was doing my PhD, also in London, because then the lab had moved and I was still finishing up experiments. So I was there for a year postdocing, finishing up my work. So up to this point, I was a stranger to synthetic biology. I'd read stuff about it a little bit. I didn't know a whole lot, but it was in fact when I was writing my PhD thesis, this was 2010, that a lot of papers in synthetic biology were coming out, and or maybe they had been coming out already, but they were catching my attention at this point, partly because I was reading a lot at this time to write my thesis, and I got very excited about it. And I thought, oh, well, what I'm doing, I was doing at the time molecular parasitology of an organism called Trypanosoma brucei that causes the African sleeping sickness. And this organism was very exciting. I was working on chromatin remodeling in this organism. But somehow I felt that SynBio was where I wanted to be, because there you could not only, study phenomenon if you want to, but you can engineer things and, do something interesting and useful. And I really liked that idea. And it would also give me an opportunity to, do both computational and experimental work. So at this point, I started looking for positions in synthetic biology. And then I moved for my postdoc to State College, to the lab of Howard Salas at Penn State. And I was there for 3 years. That was my first postdoc. Well, if I count the one in London, then it was my second postdoc. And then after that, I moved to Warwick University in the lab of Alfonso Jaramillo for another 2 years. And following that, I started my current position in Paris at the Micalis Institute. So it's been a long journey through 3 different continents. So the question that I'm often asked, where you're from, at the moment, I don't know anymore, because I'm sort of at this point, become a bit of a global citizen, if it were, or a global nomad. But right now, for the last few years, I've been situated in Micalis Institute, which is a great place to do research. I really like this place because it's a mixed unit between different institutions. INRAE that I already described, the National Lab, AgroParatech, which is an educational more engineering university, and then University of Paris-Saclay. And we have people from all these universities at the Micalis Institute. It's located in the south of Paris. So you have France and you have the big Paris region. In that red region is where is the Paris city. And we're in the southern suburb of Paris, not very far from Versailles. And the Micalis Institute itself is it's a medium-sized institute, I would say. We have about 20 research teams, 340 people. We have a bunch of themes around studying microorganisms. So we are interested in emergence control and adaptation of pathogenic microorganisms, how they interact with the environment. We're very interested in microbial food and intestinal ecosystems. How does food affect health and everything around that? And we're interested in systems and synthetic biology. And it's that theme that I'm a part of. To just give you a sense of what kind of a campus we are very green, sort of rural-ish campus. So, as I said, it's a suburb of Paris, so it's not, in the big city. You have large fields with a lot of, cows and ducks and geese and other animals that you will see on campus. And we're not very far from the Chateau of Versailles, so it's always great to, go out there, hang out in those parks and so on. So with that little introduction, which I think has lasted a little bit too long, I will jump into the topic at hand, which is looking at what I do in the lab. So my interest, as I told you, we're a multi-PI team. So of course, the team has many broad interests. They all fall within the, the general range of metabolic engineering and synthetic biology. Within that, the specific thing that I'm interested in is sort of design and analysis of genetic circuits and metabolic pathways, biosensors for RNA, for metabolites and such like. And also more recently, I've started sort of investigations into how to engineer distributed systems using phages for intercellular communication. But today, what I'll be talking about is really a part of the middle theme that we work with in the lab. And we've been working with cell-free for some time, so we have been using cell-free for sensing RNAs and metabolites. We have previously published work in what we call the metabolic perceptron, which effectively is a method of sensing multiple different inputs, and you can change the classifier for the final output just by dosing the right amount of DNA. So this was something that we published 3 years ago. More recently, we've published work in, this was in fact a project that came out of our IGEM team, where we use RNA from rosewood to basically be able to sense rosewood signatures as a way of controlling its trafficking, because it's a very highly trafficked wood around the world. And the work that I'll talk about today is more on the lines of how to sort of build systems in cell-free. And we all know that cell-free systems are, I'm trying to hide this. I'm not sure if you guys are able to see this, but, right, okay. So we know that there are many advantages to cell-free systems, and one of the key advantages that have been explored so far is that it allows you rapid prototyping. So in this recent review, in fact, from Michael Druitt's lab, there is, I've just presented a few examples here. You can use cell-free systems to produce different proteins with modifications, modified amino acids, or you can add specific enzymes to this to produce glycosylated proteins and so on. You can do pathway prototyping by mixing different kinds of enzymes together and seeing how that, which enzymes are the best ones for your substrate conversion and so on. You can also use them to study protein-protein interactions. You can express your proteins in cell-free and look at how they interact with each other, or you can use them as biosensors for nucleic acids, for DNA, for RNA, and for small molecules, as I talked about earlier. So there's a lot of things that you can do with cell-free systems for prototyping, but of course now cell-free is also being expanded to do more with it, which is more biomanufacturing, producing, personalized medicine. So you want to produce specific compounds at small scale. You can use cell-free for that. And those are some of the future avenues that I think cell-free have a lot of promise for. Right, so most of the applications in cell-free typically use circular or plasmid DNA. And there has been for many years now, in fact, this idea that, or not this idea, but sort of this realization that making large amounts of this DNA is in fact not easy. Very often you run into cloning problems if you're trying to express anything. If your plasmid codes for anything that's toxic, you will get quickly mutations, or you will get toxicity, and so you'll not be able to prepare enough of the plasmid DNA. And you will spend a lot of time cloning your construct before you're able to do any prototyping. And to fasten or to hasten this process, it has been proposed for some time that linear DNA is a good alternative because it's easier to produce linear DNA. You can simply PCR amplify using the right primers with the right overhang from a given synthetic DNA, or from even you can amplify from genomes. And once you have this PCR amplicon, you can directly use it in your cell-free reaction. So having PCR amplified linear DNA is very useful. It helps you reduce cost and also reduce time. And for that reason, it's great to use linear DNA. But there is a problem. The problem with linear DNA, of course, is that it's not tolerated well in at least extract-based cell-free systems. So if you are making a pure extract where you have, purified all the components and added them together, in the pure system, this is probably okay because it doesn't have any exonucleases in it. But if you're using crude cell extracts, and we'll come to why sometimes those are quite important to use, then you have your DNA is not very stable, and it'll quickly get degraded by the exonucleases that are in the system. In fact, in this recent review by Mark Diplinski's group, they summarized some of the ways in which linear DNA has been protected in experiments that people have already done. So, of course, a very promising approach is that you identify the most important exonucleases and you knock them out. And this, in fact, has been shown a while back. So I think in the 80s, some of the cellular extracts that were made, it was already found that mutants of RECB, for example, extracts from mutants of RECB already protected linear DNA better than wild type extracts were. Or you can remove the nucleases by sort of affinity tagging them, by sort of pulling them down and removing them from the lysine. Alternatively, you could use proteins that bind the ends of this the linear DNA and protect them. So gamma is an important protein that will bind to the exonuclease complex and therefore prevent it from degrading your DNA. Similarly, chi DNA sort of sequesters away the exonuclease complex, and by doing that, you can protect your linear DNA. Or you can use DNA binding proteins such as tough and Tur proteins that have been used in literature. They can also protect the ends of your linear DNA. The other things to do to protect your linear DNA are DNA methylation, where methylated DNA seems to be better protected against exonucleases, so then your DNA is going to last longer in the system. Or you could use modified ends of your linear DNA, so phosphorothioate links that will effectively protect against the exonucleases at the end. Or you could also do an in vitro circularization before you put this DNA into your cell-free mix, and that will protect this DNA. So, of course, many of these methods already exist. One of the problems is that in all of these sort of examples on the right, the nuclease inhibition or the LET modifications, or even with the removal of nucleases, there are all these additional steps involved that, in fact, make it more expensive, both time-wise and resource-wise, to sort of do your experiments. So we were looking for an easier way to do this, and we wanted to effectively go back to the original method that had been seen in the 80s with the nuclease knockout, and we thought, okay, let's make a strain that will, where we get rid of the exonuclease 5, which is the main exonuclease that is known to degrade linear DNA. It's encoded by the RECBCD genes. And if we delete those, this would be a great way of protecting linear DNA, and then we can go ahead and just use those lysates in the future. Now, of course, this had been done before, as I already told you, but it had been done in strains that were not the most productive strains. So it was originally done in E. coli A19, and these strains at least are not, I don't know if they are in a repository. I tried looking for them, I could not find them. And I also, at the time, we were comparing the notes from literature, and we've in fact found that there were papers that had compared protein production rates from the A19 strain and the commonly used strain, the BL21, and BL21 in fact is almost 30 to 40% higher protein production than A19, and so we decided that we'll make the BL21 knockout strains again, and we go with and we go from there. So at this point, in fact, we were so a PhD student Angelo was working on this, and one of his PhD committee advisors was Chase Basil, and we were discussing with him, and he was also interested in making these RECB or RECBCD deletion mutants. So then Angelo teamed up with Tanya in Chase's lab, and they started knocking out the RECB gene or the RECBCD gene from the E. coli BL21 Rosetta 2. That was the parental strain that we used because in the past we had used this strain in the lab, and it's quite a highly productive strain. It also contains the P-rare 2 plasmid, which encodes for rare tRNAs. So it's also great for expressing non-codon optimized genes that come from other organisms. And normally you would have to re-optimize it for E. coli, but in this case, because you have the P-rare plasmid, you can use them directly in these lysates. So of course, we went ahead and did these designs. We did land-red recombination to get rid of either RECB or RECBCD, the whole, all the three genes of the complex. And then we wanted to be sure that these cells grow fine, because there had been previous reports that indicated that although RECB seems to grow all right, RECBCD is severely impaired in its growth. So we were expecting that RECBCD deletion mutants will grow much slower, but at least in our hands, we did not find that. So there was about 25% growth reduction if we look at the BL21 Rosetta 2. And here are the BL21s without the P-rare 2 plasmid. And in both of those cases, the reduction is less than 25% in specific growth rate. So we were quite pleased that we're able to maintain these cells well. And now we can go ahead and make lysates from them and start using them for linear DNA expression. So that's what we did. We made lysates from these extracts. We optimized the buffers, and then we tested them. So this is the wild type extract, and these are the RECB or RECBCD deleted extracts. And we found that although there was a small amount of expression, about definitely more than in the wild type, the linear DNA was immediately degraded. But in these two deletion mutants, there was the linear DNA was protected, and it was giving us GFP expression. The problem was this GFP expression was not very high when you compared it to the plasmid DNA. So because we were interested in optimizing this to get the best expressions, we wanted to compare to plasmid DNA. And we saw only about 20% of expression from plasmid DNA in both the delta RECB and the delta RECBCD extracts. And then we started wondering, well, maybe, and this was something that was quite actually common in our lab and with other collaborators that we work with, that very often we'd found that depending on the exact extract, or depending on even the exact plasmid, you might need to optimize the buffer differently. And this got us wondering, well, is it possible that linear DNA would give us better expression if we optimize the extract specifically for linear DNA? In this case, we just had followed our regular protocol for buffer optimization, where we normally use a plasmid DNA to optimize our buffers for best expression, and then we use those compositions. So at this point, we were, and we have been for long-term collaboration, we have been working with Jérôme Bonnet's team, who's in Montpellier in the south of France. And there was a master student there, Antoine, who was also interested in the linear DNA question. And he started doing these linear DNA specific optimizations. So what he did was he took linear and plasmid DNA, and then he did buffer optimization like we normally do. We first optimize mg glutamate concentrations. And when he did that, he found that for the wild type extract, in fact, the linear DNA was very quickly degraded, so he didn't get a lot of expression. But for plasmid DNA and linear DNA, they were approximately similar K-glutamate, mg glutamate optimal concentrations, and also similar K-glutamate concentrations. But when he started looking at the exonuclease depleted extract, he found that, in fact, for mg glutamate, it seems that the optimal concentrations are the same, whether you use plasmid DNA or linear DNA. But when you compare the concentration of K-glutamate in the buffer, you get very different optimals for plasmid DNA versus linear DNA. And this is true in both the REC-B, in two different two different extracts that he took along. So in this work, in fact, we took a number of different extracts in these experiments, because in the past we've seen a lot of variability from batch to batch and from research personnel to personnel. So as a result, in this particular study, we did many of these experiments, both in the Montpellier lab and in the lab in Paris, and we compared results. And we, in fact, did it at slightly different conditions as well. So the Montpellier lab likes running their extracts at 37 degrees, and they use French press to make the lysates. We here in Paris use sonication to make the lysates. We run our experiments at 30 degrees. And they were using the BL21 without the P-rare 2 plasmid, and we were using the one with the P-rare 2 plasmid. So there were small differences, in the materials, but in fact, as you will see, most of our results were similar across the two labs. So this was quite comforting, because it told us that these are not artifacts of specific batches or specific people working with this. So going back to what he noticed that in fact in two of his extracts, he gets K-glutamate concentrations of plasmid DNA, range 140 to 200 or 220 in this case, and he was getting that for linear DNA. He would get it between 80 and 140. So then we wanted to ask whether this is true in our settings in Paris. And so we repeated these experiments here, and we repeated them both for the delta REC-B and the delta REC-BCD extracts. We had, in fact, two of these delta REC-BCD extracts, and we found the same behavior. So at a different temperature, a different method of lysate preparation, even a different strain. But mind you, these strains are still, so one of the strains of delta REC-BCD, and there are two lysates that we had one is delta REC-B and two lysates from the delta REC-BCD strain. And in both of them, we find that the same pattern is repeated. So when you are using plasmid DNA for buffer calibration, you find that you need sort of 140 to 160 nanomolars of K-glutamate, but you only need 20 to 40 nanomolars for linear DNA optimal. And in fact, in this case, we found that the expression that we were getting from linear DNA were equal to, or sometimes even better than what you were getting from plasmid DNA. And in this case, the plasmid DNA data comes from the buffer that has been optimized for plasmid DNA. So it's not like we're using these differences because we put it in the wrong buffer net. So we stuck with the optimal buffer for the plasmid DNA, optimal buffer for the linear DNA. And when you compare that, you can see that linear DNA is very similar levels of expression, even higher sometimes than plasmid DNA. So this was very promising. And this meant that we could make these extracts and use it for our prototyping applications and so on. But we wanted to ask a few more questions. So we were wondering, I told you that there are other ways of protecting linear DNA. So you can add supplements like GAMS or Chi and DNA. And we wanted to know if there was some kind of a synergistic effect, that we would find if we combine the two. So in this case, what we did was we used the wild type extract and we used the delta rec BCD extract. And in both of these extracts, we added either gamma supplemented. So before we did this, we already had optimized the right amount of gammas that would give us the best protein expression in our lysate. And when we compared that to in BL21 wild type and the delta rec BCD, we in fact found that there is some amount of protection that gammas and chi6 provide to linear DNA in the wild type extract. But in the exonuclease-deficient extract, there is no additional protection that's available. Already, the delta rec BCD extract is quite good at protecting linear DNA, and you get high expression from it. And this was something that we wanted to sort of also verify in another way. So what we did was here in Paris, we transformed cells with gammas expressing plasmid, and we made lysates which had pre-expressed gammas in them. And then we also found the same behavior, which was that in gammas containing extract, when you compare the wild type lysate, there adding the gammas really increased the protection for linear DNA. In the wild type extract without gammas, there was almost no expression. But when you have it with gammas, you get high expression from linear DNA. But in the exonuclease-deficient extract, there is no added advantage of the gammas pre-expression. In fact, it seems to be that gammas reduces the expression from these extracts. What we curiously also noticed was that adding the gammas, in fact, reduced the performance of the plasmid DNA in these extracts. So I think it's something to do with the gammas protein taking away too many resources before the extract is produced, and possibly there is fewer resources available or fewer ribosomes available in the system for the expression of our gene of interest. At this point, so we were pretty sure that adding these additional things does not help expression from linear DNA. So we wanted to, but we were still quite curious whether, I mean, we were quite puzzled as to why low K glutamate concentrations were giving us higher expression. So we wanted to explore that and I missed this slide. Gosh, I'm sorry. All right, I think I'm missing a slide, but that was weird. Maybe I will. Sorry. All right, I will continue and I will show you that that slide later. Right. So, okay. So we saw that adding additional supplements here is not going to protect the linear DNA any better. We also wanted to ask whether, you know, there is an additive effect if we add modifications to the ends of the DNA. And here we used a number of different modifications. So phosphorothioate, phosphorylation of the, and all of this was done essentially by, you know, ordering modified oligos with the modifications at the ends, and then we PCR amplified this and we compared. So in the wild type extract, you find that at least the phosphorothioate modification does give you some additional protection over unmodified DNA in the wild type extract. But when you look at the delta rec BCD extract, there is no additional protection that comes from these modifications at the ends. So this told us that we already had the optimal that we were looking for, and we didn't need to make, we will not improve our expression level by making these additional modifications. So at this point, maybe I will go back to, really, I really wanted to show you this data, so I'm just going to do it here. Okay, I'm going to switch my screen share and then I'll come back to that. Sorry about this. Right. So I hope you can see my current screen. Are you able to see my current screen?
Yep, we see the ACS and bio paper. Excellent.
So we were actually quite curious as to what was going on. Why was it that our extracts with the low low K glutamate were protecting better? So we wanted to know whether this was something to do with better protection of the DNA itself, or whether there was something else that was going on here. So we dug into the literature, and in fact, we were able to find that exonuclease-9, I believe, is sensitive to potassium ion concentrations. And we wondered if there was this additional exonuclease, which was still in the system, and by changing the potassium glutamate, we were somehow changing susceptibility to that. So in order to do this, we ran a cell-free experiment where we have the wild type lysates, low K glutamate and high K glutamate. In that case, you of course see that there is no expression because the linear DNA is very quickly degraded. In the other extracts, the delta rec BCD and the delta rec B, you do see high expression. But here then we quantified the amount of DNA left at different time points in this reaction. So at 0 time point, at 1 hour and 8 hours. And again, with the wild type, you see there was a quick degradation of this DNA. But in the extract with the delta rec B or the delta rec BCD, you see that there is a reduction in the amount of DNA as time goes by, but in fact, there doesn't seem to be any substantial difference between the low K glutamate and the high K glutamate settings in any of those two lysates. So that told us that it's not really the linear DNA that is being protected better. In fact, what is going on is, so at this point then, we were quite puzzled and we started digging the literature more. And in fact, what we found was that there had been previous literature evidence that K glutamate tends to destabilize the RNA polymerase on the promoter. So this is so this was something that we did not know before, but as a result of these results, we were able to dig back and then find, in fact, 2 papers that had looked into this, where they were doing in vitro studies where they would express the RNA polymerase from E. coli, and then they would change the buffer composition, and they found that when you have a high K glutamate, in fact, the native RNA polymerase from E. coli is destabilized, and we think that explains why we get lower expression when you increase the K glutamate concentration. So I will stop this share and I will go back to the original presentation. Right. So now that we had a notion of why our system was working, and we had tested it through different lysate preparation methods with different backgrounds of the parentals, even different temperatures. We wanted to see whether we can use our new lysates to do some rapid prototyping. And in this case, we chose a small library of 11 PCR assembled toll switches. So here we started with a published trigger RNA from Green and Tal paper from 2014, and it's and the riboswitch that was originally published in the paper. And then we made many modifications to the RBS region in this riboswitch. And effectively, we produced this trigger RNA and we added it with and without. So we have the experiment where we PCR amplify the riboswitch, we add the RNA or we don't, and we see whether the trigger RNA's presence activates the riboswitch. So in this case, we found that although the best fold change was truly the original published riboswitch, the toll switch, gave us that best fold change. In fact, our variant J61106 version of the RBS, in fact, gave us lower background expression. So, and in fact, had a slightly higher fold change, although its dynamic range is lower. So depending on the application, one may want to go for the original RBS or this one. But it at least demonstrated to us that this is a great way now to be able to quickly assemble a lot of different ribose switches, PCR assemble them, and just put them into the lysate and test out different ribose switch designs. All right, so now that we could do this for sensing RNAs, we also wanted to use that for identifying better transducer enzymes. So I will just quickly define what transducer enzymes are. So in the lab, we are interested in sensing metabolites, and sometimes you don't have the right transcription factor for the metabolite that you're interested in. And what we use is something that we call sensing enabling metabolic pathway, which is essentially a way of converting a molecule that you cannot sense, in this case, hippurate directly, into a molecule that you can sense, benzoate. And you do this using an enzyme that we call a transducer enzyme because it transduces one kind of signal to another. And now you have a transcription factor, BenR, that can sense benzoate. Therefore, indirectly, you can sense the amount of hippurate in the system. So we already had in the lab a particular version of the HIP-O enzyme that converts hippurate to benzoate. And we wanted to test whether other variants of this enzyme may be better. In order to do that, we took plasmid versions of the BenR transcription factor and the reporter plasmid, but then we had different variants of the HIP-O enzymes that were PCR amplified and added into the system to see which one performed better. So in this case, we again use a small library of 4 homologs. Our original one came from Campylobacter jejuni, so that's this one here. And then we tried 4 others from 4 different organisms. And in fact, we found that the one from Helicobacter felis and Campylobacter coli gave us even better fold change than the original one that we had in the lab. So this was quite promising. And so we know that in the future, when we want to sense hippurate, we can use the HF enzyme and not the CJ one. So with this, I hope that I've convinced you that linear DNA is quite a great way of, in fact, rapidly prototyping your system. It may also be, it saves you time, of course, and for us, what was, as I've already said, what was very interesting was that it told us that you need to optimize your buffer specifically for the settings that you want to run in. So there may be plasmid to plasmid differences, and there are definitely linear DNA to plasmid DNA differences in terms of the optimal buffers to use when running your reaction. So we have a workflow here of how to, this is, I mean, to the cell-free audience, this is something very common. You will understand exactly what's going on here. But we presented this workflow, and then we have, in fact, we published earlier this year the ACSA bio paper, and we recently published a more detailed protocol which lists out step by step how to prepare these cell-free lysates and linear DNA to use for rapid prototyping. The materials to use, basically the different knockout cell lines are on Abgene, and the references are there in both the papers. So if you're interested, you can get them from there, or if you have questions about them, you can drop us an email. So I'll just quickly, sort of before I wrap up, sort of summarize the main points. So we showed that exonuclease-deficient extracts improve expression from linear DNA. We also showed that when using native E. coli transcription, cell-free buffers should be specifically optimized for linear DNA, and that the optimal settings from plasmid DNA do not work or do not work as well. This is specifically true of native E. coli transcription, as I told you earlier, because it's so sensitive to K-glutamate concentrations. Perhaps with pure system, or when you're using a T7 promoter, perhaps these differences may not be as relevant. Also, we were able to get these results in different settings, so different genes, different temperature, different DNA concentration, multiple batches. So I think overall we tried 9 different batches in our experiments, and we were able to reproducibly show that the buffer needs to be optimized differentially, and we were able to use our system for rapid screening of RNA switch designs and metabolic enzymes. with that, I'd like to thank all the collaborators. This was quite a collaborative effort between different places. So we have the team in Paris, the PhD students, Angelo and Paul, and more recently, postdoc Manas, who's taking this work forward now. Of course, the team leader, Jean-Luc Folland. In Montpellier, we have master student Antoine and Paul, the PhD student, who is in fact a shared PhD student between both the labs. So he spends some of his time here and some of his time in Montpellier. And then all the other members from CBS Montpellier and Jérôme, as well as Tanya and Chase from the Helmholtz Institute in Würzburg. And thank you all for your attention. And I'm happy to take questions. Right.
Thank you so much. That was a fascinating talk. There is a lot of questions in chat. Can you see chat?
Yes, I can see them now.
If you could read the question out loud before you answer, that's going to be great for the recording.
Yeah, right. The campus is gorgeous. Thank you. I agree with you. Could you speculate why linear DNA needs different buffer composition? So this is, I guess you asked this question early on when I showed those results, but I think we really think it needs different optimization because the RNA polymerase complex gets destabilized at higher K-glutamate concentration. And perhaps, I mean, it's not something we will test this. It's not something we've tested so far, but maybe when you use T7 promoter, you won't see such dramatic differences in buffer optimization. Sorry. Right. We thought too that gamma was inhibiting the yield. We thought we were doing something wrong. No, we saw that too. So whatever you're doing, it looks like it's a common result. I think it's to do with the fact that, so in our case, so this is exactly something we don't know whether it's because of the chloramphenicol that's the plasmid is being maintained on, or is it, oh no, I think gamma is an ampicillin plasmid. So we don't know if it's just the antibiotic or is it simply the load of expressing gammas. We know that when you add the gamma plasmid in there, these cells grow really slow. So I can imagine that the total, the effective yield from these lysates is lower, which is what we in fact saw.
So I just want to add something to this. When you were talking about it, I was thinking about it. We actually see decreased yields with GAMS even when we add GAMS as a purified protein.
Oh, really?
And that was really puzzling to us. And we actually had samples of GAMS that we made in-house, and we had some original samples from Richard Murray's lab. And in our hands, GAMS was decreasing yield, whether we express it from a plasmid or whether we're using it from purified protein. So there's something funny going on, and I'm glad to see others have problems with it too, because that's not just us being incompetent.
Yeah, no, I mean, for us, we saw this. So we saw this mainly when we pre-expressed GAMS. Mm-hmm. When we purified it, in fact, because now I don't know if it's something to do with the optimal amounts of GAMS to add, because in our case, we first optimized it. So we picked the one where we got the best protection. And perhaps that's why in the final data, we don't see it. Maybe that could be a solution. If you have purified GAMS, you first find the optimal for it. Because perhaps there are batch-to-batch differences even in the production of GAMS.
Yeah, that's a very good point. We didn't optimize it. We used the published concentration, and we switched to using a knockout extract just like you guys now. So
yes, I saw the Akabi paper. So
so we don't use GAMS anymore either. But I'm glad to hear that other people were struggling with it too.
I mean, since we're talking about that, can you do you see? I mean, did you ever compare plasmid DNA to linear DNA with? Because I noticed that you use T7 promoter, and there what kind of expression levels when you compare plasmid versus linear?
We still see, even in our knockout extract, we see slightly lower yields with linear than with circular, but they're within the same order of magnitude.
Okay.
They're not too far off, and there could be batch-to-batch variability too.
Right. Is there a relationship between the length of the linear DNA and yield? or will old length templates perform similarly? That's a very good question. So we did not, so in this context, we did not test this systematically, but because of differences that exist in the two labs and the plasmids they use, we know that we have differences in the flanking regions. And there have been previous work that shows that the longer the flanking region, so between your promoter and the terminator, if you add more spacer DNA on the side, this tends to protect linear DNA better. And so I think the answer to that is yes. There is a relationship. Specifically, it's been studied for the flanks. Whether, if you have, multiple coding regions in it, I don't know. We've never tested multiple coding regions in the same linear DNA. But definitely for the flanks, there is a relationship. Is RecBCD the only DNA nuclease in E. coli, or are there different ones that could also be knocked out to increase yield even further? There are other ones. As I told you earlier, we were suspecting this Exo11 because it's K-glutamate dependent, but it seems that for linear DNA, the most important one is RecBCD. And so if you knock that out, you're relatively safe, although we have not tried to knock out any others, so I cannot say if it would help. Have there been work done on the expression of linear DNA templates and other types of extract, like yeast to mammalian? I actually don't know. I don't know. I'm not, I don't remember reading a paper, but then I have not really kept kept up to date with the yeast and mammalian cell-free literature. So perhaps there are gaps in my knowledge about it. Wonderful talk. There is an upper limit to how many different linear DNA templates can you use in TXGL. In general, I think there will be an upper limit on how much expression you can get out of linear DNA. I did not talk about this here, from any cell-free system, linear DNA or plasmid. I didn't talk about this here, but in the metabolic perceptron work that we did earlier, whenever we were adding multiple plasmids, we had a simple, Hill kinetics model to explain the expression behavior based on the amount of DNA that was added. And when you added one piece of DNA, one plasmid, it was fine, and the model was all predictable. But the moment you added a second one, there was resource competition, and this means that the other one will get expressed lesser. So I am pretty sure the same rules will apply for linear DNA templates as well. If you add more and more templates, you will probably see that individually, each one of them will get expressed lower. Could you please comment on variability between batches of the extract? Is higher or lower variability of linear versus circular DNA templates? That's a very good question. So in general, we do see a lot of variability from batch to batch, from individual to individual, and in a previous work, which one of the PhD students, Paul, in fact, both Paul and Angelo were involved in this work. It was done by postdoc Olivier Borkowski in the lab, where they were looking at they were looking to optimize buffer compositions, and they took 11 different components and then used a machine learning method to optimize this. And what they found was that there are ways to essentially, so once they did this 11 component analysis, they were able to whittle it down to 20 buffer composition. This is a Nature Communications paper, Borkowski et al. From a few years ago, so you should be able to find that. But once you have the data from these 20 different buffer composition, you can reduce the variability from batch to batch extracts by essentially tuning your buffer composition based on this data learned from these 20 points. So the model can learn on these 20 points and then tell you so you say you maximize it for this lysate, and now I want to get similar expression from the other lysate. So you could do that, but I think there will still be the challenge that, what if at some point you end up getting a better lysate? Will you want to tune it down to match what you have been using in the lab before? Or will you want to get the maximum yield? And I think that makes this quite hard when you're using, cellular extracts. I think the variability is probably lower in pure systems, but we don't use pure systems a lot, so I don't have first-hand data to talk about here. What is the relationship between performance and the desired protein size? Good question. We have not formally looked at this relationship between the size of the protein, but I would imagine again that if you have a larger protein, your maximum expression will be lower than for a smaller protein. But this is a guess simply based on finite resources in the system. Are all the promoters use sigma 70? If so, did you try T7 promoter to see whether the results would be different? In this work, we did not try T7 promoter. We used sigma 70 promoter, and historically in our lab, we have done this because we arrived, to cell-free more as a biosensing mechanism. And if we want to sense specific metabolites using transcription factors, then these transcription factors will have to use the native promoters, which often will go through the E. coli native RNA polymerase. So if you want to have a T7 promoter activated by a small molecule, then there is a whole lot more engineering involved there. So that's why we always use native E. coli promoters. I can't share specific examples, but there is minimally a molar relationship between linear DNA length and yield. Equal mass of two different length DNAs will result in less expression of the longer DNA because this molar quantity is reduced. Yes, yes. So all the data I presented today, we always aim for, when we make comparisons, we're always aiming for the same molarity. We never look at the concentration in nanograms per microliter. So you're right, of course, if the molarity of the DNA is lower, then in any way you have fewer templates of this DNA. So you will have a lower yield. How does expression level from linear to plasmid DNA compare to expression without transcription, just using purified mRNA? This is not something we have specifically tested. We did not test this. The only time we used purified RNA in these experiments was when we added the trigger. The trigger we expressed in an in vitro T7 reaction and purified and added that. But here we did not add pre-expressed mRNAs into the system to test. But this is a good idea. I think this is something we should do.
I think that's all the questions you had.
Thank you.
I was going to ask a couple, but people asked them for me already. So I think I'm all set too. Thank you so much. Thank you so much for a great talk, and thank you for answering all the questions.
Thank you. It's been a pleasure talking
thanks
to you guys.
Thanks everyone else, and I hope you all have a great week, and see you next week.
All right. Bye-bye. Have a great week.
Bye.